Good morning. This is Dr. Ari Lawal from the Department of Biochemistry, College of Medicine, University of Lagos. We're going to be looking at the brain chemistry and neural signaling today. What you hear, you forget. What you see, you remember. What you do, you understand. This is attributed to Confucius in between 551 to 479 BC. There is no erasing in the brain. Now, if a human reads, the sensory, motor, and central nervous system all come together to pass on the message through a neuron, through synaptic vesicles, with the aid of neurotransmitters across the membranes to pass the message down. The brains and computers, the brains, our brains are able to perform fewer operations per second compared to the computers. However, the brain has more units than the number of processors you find in the computers. There are about 10,000 connections in the brain, while the computers have just about 4 connections. The brains are fault tolerant. They don't crash unlike the computers. The brains also evolve while the computers are designed. The brain is able to learn while the computer has to be programmed. How does it all work? The molecules such as neurotransmitters, biomolecules, they all come together through synapses, build up synapses. The synapses are there in the neurons. The neurons come together to form larger networks, maps. And then you have the systems, different systems in the central nervous system. So the level of investigation you have from the central nervous system, you look at the different systems within the central nervous system, the networks, the array of neurons that come together uh, via their synapses, and then the molecules, the smaller molecules that interact, such as the neurotransmitters. Now, single cell behavior, most protozoans have no nervous system. They're able to swim or move towards chemical or field. They're also able to avoid harm. They move, they are motile, and they move away from harm. They are also able to reproduce using chemical messenger proteins. The membrane, this is a typical membrane showing the point of interaction between a cell and its external environment or the stimuli. Now, the earliest nervous systems were found in hydra, jellyfish, and the rest. The basic neural cells that you find in the nervous system are the neuron. The neuron, also referred to as the nerve cells, are differentiated into 3 types. The first is the sensory neuron that takes messages from the body to the central nervous system or the brain, while you also have the interneuron that links the sensory neuron and a motor neuron, which is tasked with passing on the message from the neurons to the central nervous system. Now, neural processing, the neurons or the nerve cells have what you call the cell body. The cell body, also referred to as soma, is tasked with metabolism, protein synthesis, cell sorting, synthesis of lipids, and the rest of them. The cell body has its own nucleus, it has its own endoplasmic reticulum, mitochondria to generate energy. The dendrites are the input structures that you find close to the cell body. They receive inputs from other neurons, and then they also perform special temporal integration of these inputs. They relay the information to the cell body, while the axon is known to be an output structure, which is a branching fiber that carries the message from the cell body to other neurons down through its terminal. This is um, the diagram showing the dendrites, the nerve cell body, its nucleus, and then the direction in which messages pass down the axon to the axon terminal, from which messages would live through a synaptic cleft to a postsynaptic neuron down there. Now, the synapse is the site of communication between two cells. It is formed when an axon of a presynaptic cell connects with the dendrites of a postsynaptic cell. The synapse shown here has a space referred to as the synaptic cleft. The axon of a presynaptic neuron contains vesicles has vesicles, old vesicles, which contains neurotransmitters. These neurotransmitters, when they are released, can be passed on to their receptors on the dendrite of a postsynaptic neuron, as shown here. A synapse can be excitatory, or it could be inhibitory. Arrival of activity as an excitatory synapse depolarizes the local membrane potential of the postsynaptic cells and makes the cell more prone to firing, or more prone to being able to pass on message. So it usually connects on dendrites. Now, arrival of activity at an inhibitory synapse, on the other hand, hyperpolarizes the local membrane potential of the postsynaptic cell, and it makes it less prone to firing. Usually, this connects on cell body. So the greater the synaptic strength, the greater the depolarization or hyperpolarization, as the case may be. Neural communication within the cell. Transmission of information occurs within and between nerve cells. Now, information must be transmitted within each neuron, and it must also be transmitted between neurons, as shown in the diagram. The membrane surrounds the neuron. It is that barrier that delineates the nerve cell from its environment. It is composed of lipid and proteins. The lipid bilayer, as well as intervening proteins embedded within the lipid bilayer. These proteins are known to have enzymatic function, receptor function, transport functions. Some of them are channels, some of them are carriers, some of them are pumps that pump ions, and the rest of them. Now, this membrane bilayer, this is a typical fluid mosaic model showing the transmembrane proteins. They are integral membrane proteins that are able to traverse the whole lipid bilayer. Well, you also have glycoproteins and I can see glycolipids that are cell adhesion materials, which allows for the cell to interact with another cell. Now, at the resting potential, every cell has what is referred to as a resting potential. It's an electrical charge across membranes. The inner of the membrane, the inside of the membrane, is majorly negatively charged, while the outer part of the membrane is majorly positively charged. So the difference between the inside and the outside of the membrane is a membrane potential. The resting potential is the point when the cell is not firing. The resting potential is a 70 millivolt difference between the inside and the outside of a cell. So the resting potential of neurons are about minus 70 millivolts. So the ions and the resting potential, majorly ions are electrically charged molecules. For instance, you have the sodium ion, the potassium ion, and the chloride ion. They are found on either side of the membrane. The resting potential exists because ions are concentrated on either sides of the membrane. Sodium and chloride ion are found outside the cells, while potassium and organic anions are found inside the cell. To maintain the resting potential, sodium ions are actively transported. They're actively transported with the aid of energy to maintain the resting potential. The sodium potassium pump is a membrane protein which exchanges 3 sodium ions for 2 potassium ions. So you have 3 sodium ions are pumped out while 2 potassium ions are pumped in. This is the molecular organization of the sodium potassium ATPase, a model for the catalytic subunits showing the transmembrane. So the sodium potassium ATPase has domains through which sodium ions will bind on their way out, while it also has domains for potassium ions to bind on their way in. Now, integration of information. The PSPs are small and individual excitatory postsynaptic potential will not produce enough depolarization to trigger an action potential. However, inhibitory postsynaptic potentials will counteract the effect of excitatory PSPs at the same neuron. Summation means the effect of many coincident IPSPs and EPSPs at one neuron. If there is sufficient depolarization at the axon lock, an action potential will be triggered. Neuronal firing, the action potential. The action potential is the rapid depolarization of the membrane. Since we know very well that the membrane is polarized with more negatively charged ions within than outside, while we also have more positive ions on the outside than on the inside. So the neuronal firing starts at the axon hillock and passes quickly down along the axon. The membrane is quickly repolarized to allow for subsequent firing. That means as it gets depolarized, you have less negatively charged ions within, while the membrane is quickly repolarized to allow for subsequent firing. Repolarization would imply allowing some of the positively charged ions get back into the cell. And before depolarization, more positively charged ions are found outside, while the more negatively charged ions are found inside. Now, action potentials and rapid depolarization. When partial depolarization reaches an activation threshold, voltage-gated sodium ion channels will open and then sodium ions will rush in. So basically, when sodium ions rush in, the membrane potential would change from about minus 70 millivolts to plus 40 millivolts. The sodium ion and the negative ions, and the positively charged ions are found outside, while the negatively charged ions are found inside. The sodium ions here rushing in, and then you have the negatively charged ions and the positively charged ions being swapped. Depolarization causes the voltage to rise, and then repolarization brings it down to its former resting potential. And if there is hyperpolarization, if repolarization occurs, it becomes hyperpolarized. Now, action potentials, repolarization. Sodium ion channels close and they become refractory. The depolarization triggers opening of voltage-gated potassium ion channels. Potassium ions will rush out of the cell, repolarizing and then hyperpolarizing the membrane. So the action potential is all or none. it is always the same size. Either it is not triggered at all, e.g. too little depolarization, or the membrane is refractory, or it is triggered completely. Now, conduction of the action potential. Passive conduction will ensure the adjacent membrane depolarizes so that the action potential travels down the axon, but transmission by continuous action potentials is relatively slow and energy consuming. A faster and more efficient mechanism has evolved. That's the saltatory conduction. Myelination provides saltatory conduction. In synaptic transmission, information is transmitted from the presynaptic neuron to the postsynaptic cell. Chemical neurotransmitters will cross the synapse from the terminal to the dendrite or soma of another cell. The synapse is very narrow, so transmission is fast. The structure of the synapse, an action potential causes neurotransmitter release from the presynaptic membrane. Neurotransmitters then diffuse across the synaptic cleft. They bind to their receptors on the postsynaptic membrane, altering the membrane potential in that process. The calcium ion causes vesicle membrane to fuse with the presynaptic membrane. The vesicle contents are emptied into the cleft via a process of exocytosis. Neurotransmitters will then diffuse across synaptic cleft. Ionotropic receptors. Ionotropic receptors, ligand-gated. Synaptic activity at ionotropic receptors is fast and brief. Acetylcholine works in this way at nicotinic receptors. Neurotransmitter binding changes the receptor's shape to open an ion channel directly. When a neurotransmitter binds to its receptor, it allows for a closed ion channel to open, and when it opens, that allows for the ions to flow in or out, as the case may be. While metabotropic receptors, on the other hand, are those receptors when neurotransmitter binds to them activates a G protein. The G protein then splits, or the alpha subunit of the G protein breaks away and binds to an ion channel, and this allows for the ion channel to open, after which the ions are able to enter the cell and produce the postsynaptic potential. On the other hand, the molecule or neurotransmitter can bind to the receptor, and the receptors can then activate the G protein. The G protein has its alpha subunit breaking away, and thereby activating enzymes such as the adenylate cyclase, which produces a second messenger. The second messenger then binds to an ion channel, and by binding to the ion channel, it allows for the ion channels to open, and the ions flow in to produce the postsynaptic potential. Opening of the excitatory postsynaptic potentials. Opening of ion channels, which leads to depolarization, makes an action potential more likely. So hence you can have an excitatory postsynaptic potential. Inside of postsynaptic, cell becomes less negative. As you have a depolarization. So the sodium ion channels would open and allow for influx of sodium ion, calcium ions to flow in, thereby reducing or making the inside of the cell less negative. Now, inhibitory postsynaptic potentials, on the other hand, ensures that during the repolarization, the opening of the ion channels would lead to hyperpolarization, that's repolarization, a more intense repolarization, and this makes an action potential less likely. hence becomes inhibitory postsynaptic potential. Inside of the postsynaptic cell becomes more negative, more than its resting state. So potassium ion channels are open to allow for potassium ion to go out, while chloride ions flowing and the inside of the cells become more negative. The postsynaptic ion motion. Ion movements during postsynaptic potentials are such that when the molecules or neurotransmitters bind to the postsynaptic binding site or the postsynaptic to its receptors on the postsynaptic cells, sodium ions will flow in. The influx of sodium ion will cause depolarization. That's an excitatory postsynaptic potential that will allow for action potentials to move, or the stimuli to move, while the efflux of potassium ion causes hyperpolarization. This is inhibitory postsynaptic potential. The influx of chloride ion also causes intense hyperpolarization. That's also an inhibitory postsynaptic potential. While the influx of calcium ion would activate enzymes. Requirements at synapse. For a synapse to work properly, 6 basic events need to happen. One is the production of neurotransmitters, and these are stored in the synaptic vesicles. The release of these neurotransmitters on change in ion concentration across membrane would lead to the release of these neurotransmitters into the synaptic cleft, and then the binding of the neurotransmitters to their receptors on the postsynaptic membrane via a lock and key approach. Then on the postsynaptic cell, there will be a generation of a new action potential, after which these neurotransmitters that are being used will need to be removed from the synapse, either via reuptake or via degradative processes, such as you have acetylcholinesterase degrading acetylcholine into acetate and choline. Thank you.